Okay, so it's time for our keynote. So thanks very much to our speaker, Tongli Wang, who is speaking, is very patiently held on for us from Canada. And we've got our, yes, we've got our timing right, I think, which I hope is good for you. So thanks very much for waiting for us. We were just wrapping up a few interesting questions from the session. And now, please, can I hand over to you for our keynote presentation.
Thank you very much. Can you hear me clear?
Yes, very well.
Great. Okay. Thank you very much. Good afternoon. And thanks for the organizer to give me the opportunity to share my thought and some studies of our group in climate change mitigation and adaptation. Here is my talk, using forest genetic resources for climate change mitigation and adaptation. Okay, here's the outline. I'm going to talk about climate change impact and the potential genetic solutions, and the climate niche models, transfer and the response functions, and the landscape genomic approach, and a summary at the end. Let's start with the first one. So the climate has a big impact on forest trees, no matter change or not. So first, climate is a major environmental factor affecting the performance of our trees. So it has an environmental effect. And it is also a major evolutionary forces driving speciation and local adaptation, which results in among population variation in trees. So it has genetic effect. That's why different forest types are growing under different climate conditions. In other words, the nature has created different forest types for different climate conditions. Similarly, at the species level, different species are occupying different climate conditions. So we can also say that nature has created many tree species for different climate conditions. At the population level, climate also a driver driving the among population variations, create genetic clines for the widespread species, like lodgepole pine, as I showed in the figure. So the species can grow, reproduce, and grow wild within their climate niche, within the range of climate conditions. We call that climate niche. And within that, there are variation among population variations. So through long term of evolution adaptation, species are adapted to their local range. And the climate change is causing a mismatch between the climate that has been adapted to and the climate those trees will need to experience in the future. So for the trees left outside of their suitable climate conditions, they will compromise their productivity and become vulnerable to insect diseases and the natural disasters. On the other hand, there will be new area become available for the species, but due to the limited rate of migration, and it will take the trees may not likely able to catch up with this change. Here, the aspen, large-scale aspen dieback, probably due to the, is a good example of those climate mismatch. So the research questions about the climate mismatch and the possible solutions. Many people are interested to study, to understand the evolutionary mechanism of the trees in adapting to the changing climate, or specifically the mismatched climate. This is an important aspect to understand the mechanism. However, it is a completely difficult question. It will take quite a while to get there. And thus, it's not likely we can take control of the evolutionary process in near future. Many are also interested to breed more adaptive trees. This can be through conventional tree breeding programs, and it can be done. People are exploring the approach in genetic modifications, and also epigenetic approach as a merging new way to improve tree for certain trees in a more quicker way. Just like a study shown a moment ago, showing the Norway spruce, this kind of maternal memory is kind of epigenetic phenomenon. And that many people also interested to study how to help the trees to match their suitable climate conditions. So nature has created many species and the genetic clans for different climate conditions. This to help the tree to match their climate condition probably be the most effective approach to mitigate and adapt to climate change. However, assisted migration is still facing resistance from the strong idea of using local and the native species. Many people don't accept that. However, we need to be aware that the local species or local population, they adapted to their local climate, not local people, not local topography. So if the local climate move away, then the species still there is not in their local climate condition anymore. And thus, the matching their local climate should be considered as using local species, although we need to move to different locations. So in a way, yeah, using to match the climate for species is we can consider as using climatically local species or populations. Climate is changing when our conception need also to change. So the mismatch can occur at two levels. At a species level, here horizontally shown, is the climate niche, the mismatch. And vertically, it shows among population variations. So even within the climate niche and among populations variation, and the population within their climate niche may not in their optimum climate conditions anymore. So, the climate matching need to be happen also at the two levels. At the species level, we use climate niche modeling. At the population levels, we use transfer and response functions. Let's start with the climate niche models. So for species, given species, if we can build a climate niche model, then we can predict the spatial distribution of the species. Then we can also plug in the future climate to project their suitable climate spatial distribution of the suitable climate conditions for future climate. After laying down these profiles, it will help us to understand how the natural population responds at different section of this profile. So there is a leading edge and the overlapping edge, which is good for adaptation, and the lagging edge, which the populations are in danger. So this can provide the basis for us to formulate conservation and assisted migration strategies, adaptational strategies. So for the leading edge assisted migration, probably the major approach. For the overlapping area, the in-situ conservation assisted migration at a population level is the right approach. For the lagging edge, ex-situ conservation and introduction of new species should be considered. Some facts about climate niche models. So a strong relationship between the realized niche and the climate conditions is the basis of the climate niche models. The assumption is that the realized niche is a result of long-term adaptation and the interactions with other species during the past. And the major challenge for the climate niche model is the model accuracy. So this kind of model is relatively easy to build. The occurrence, which based on the occurrence data, which is easy to obtain. And also there are available algorithms to use. And so to build an accurate model is the key. And also, even you have an accurate model, the future climate is uncertain. So the future projections naturally is uncertain. And another issue is what we can predict is realized niche model. Realized niche is that fundamental niche. The limitation of the realized niche is that even you find the trees are left out of the realized niche, but they may still, within the fundamental niche, they can still survive, grow well. So it'll be good. We can also predict fundamental niche. So this is a limitation. Although those approach has been widely used, but it faces a lot of criticism. For example, the rate of migration, interaction with other species, impact of pests, diseases, interaction with microbes, and among population variations. However, those criticisms are based on how we interpret the model predictions. If we say our model prediction is species distribution model, which has been used by many people. Most people are saying this is a species distribution model. So in that case, all those criticisms are valid. In fact, the model we built in this approach is ecological model. If we say that this is a ecological niche model or climate niche model or bioclimate envelope model, and which predict the suitable climate conditions instead of the species distribution per se. So in that case, most of those criticisms are not valid. Only one is the amount population variation indeed is not reflected by this kind of models. And another issue exists for this kind of model is the extrapolability. So people say, although you can build an accurate model, but can that be extrapolate to future climate conditions? That's a question. And this study shown here is apply a smart way to validate this. They use lodgepole pine, Douglas fir. They use the Douglas fir occurrence data in North America, the native range, to build a model. Then to validate model, use the occurrence data from Europe, because this species has been successfully introduced to many countries in Europe. And their results are disappointing. So they found only a small fraction of the areas where Douglas fir successfully introduced are predicted to be suitable. So their conclusion is the SDM can be misleading. When I see this paper, I also done this modeling. And my results showing that the suitable range can be successfully predicted. When I compare with the occurrence data from the Europe for this species, and the matching rate is nearly 90%. So I have a different conclusion. So this model is highly accurate. So what was the problem behind it? So I followed the original paper, and I used scale-free data. I will come to that later. But the paper, that study used 4x4 graded data from world climb. So I did the same switch it from scale-free data to graded data. My results, the accuracy, model accuracy reduced dramatically, but still higher, but still, yeah. So the difference between these two kinds of climate data is substantial. So the grid data and the scale-free data, which reflect climate conditions for specific locations. Here, the grade, only one value for each tile. In the mountain area, 4 kilometers grade, the climate can differ several degrees for mean annual temperature. And we found a similar situation while we're modeling the ecosystem classifications in BC and Canada. And if we use the graded climate data, this is the ecosystem. The left is the ecosystem mapped. Here is our predicted on the right. So you can see the accuracy is pretty poor. But if we use the scale-free climate data, and the prediction accuracy can dramatically improve. So then this question, where to get the scale-free climate data? So for North America, and climate NA we developed. We also developed a version for Europe, and last year we published a paper on this. Several colleagues have used this model. They found that the effect is very good. And then now this Far East project developed a new version, our version using the same approach. So now come to the transfer function, transfer and the response function. So we mentioned that even within the climate niche, the population variation, amount variation is substantial. We also need to care the mismatch at the population level, but using transfer and the response function. However, those functions require provenance trials. Here I use the lodgepole pine provenance trial as an example. And this trial was built with 140 provenances, range-wide, and tested at 60 test sites. Very comprehensive provenance trial. And all the populations many populations are tested at each test site. So for each test site, the climate conditions are considered the same. For that, we can build a transfer function based on the transfer distance of the seed source in terms of climate. This here is the mean and the temperature, and they use height as a dependent variable. We build this transfer function to reflect the transfer distance on the performance. And we can also build genecology function. Instead of using the transfer distance, we directly use the source climate as the independent variables. And the results are about the same. But the genecology function is a seed source explicit. So a transfer function or genecology function represent a local adaptation because they are tested under same climate conditions. All the differences are due to their genetic differences because they are from different climate conditions. Those functions work well for the size tested, and because the same set of population, if you plant at a different test size, the function can be different. So we build all the genecology functions for the 60 test sites. Here I show 6 of them. And because each function only represents for the test size, and to generalize this transfer function, a general transfer function was proposed. And in that, you pull all the data across size into one and calculate the transfer distance and the related to performance. And this model performance is generally poor because the feature at the individual size are all canceled out each other, not all, but largely cancel out each other when you pull them together. So many general transfer function has been built for different species, and in general, they can explain about 30% of the total variation, which is not very good. On the other hand, because each populations are tested at different environment, different test sites, we can build a response function. So we use the site climate as independent variables and the tree height as dependent variables to build this relationship. The response function represents environmental effect on the performance of the population. Same genotype, different environment. So those relationships represent the environmental effect. It also reflects plasticity of the population over a range of climate conditions. So same genotype, different performance. This is a plasticity. The response function is more straightforward for climate change adaptation because the climate change is changing the environment, and the response function is reflecting the environmental effect. So it's a perfect match. However, the response function requires many test sites under a wide range of climate conditions, which is often difficult. So in many cases, for many species, they have a provenance trial only with a few test sites. This is very common. So in that case, it's not able to build a response function. And if we compare the response function and for each population, put them together, we can see that the populations for lodgepole at least from the north, and it will generally grow slow no matter where you plant them. So they are adapted to a slow growth for harsh environment. The one in the south do not perform very well either, because in the south, other species push them to the poor conditions. And from the central distribution area, it has the best performance. So in that regard, at the beginning, people think, okay, the productivity in the south will reduce in the future climate, but in the north, it will increase. So it will compensate the loss in the south. But from these results, we can see that the north, you cannot count on them to grow faster than the one from the south. So we have response functions and genecology functions. Response function, each function represents one population. The genecology function, each function represents one test size, but we have many populations, many test size, planting size, and so those functions has limitations. So we put them together and develop a universal response function. And here's a simplified version with one climate variables. So this allows us to predict performance of any population or any provenances at any test size. It's a continuous one. It also has an advantage to determine the genetic effect versus environmental effect, because they are in the same function. We can use take the first order derivatives to reflect the rate of the change. So we can use this function to predict if we use local seed for different climate conditions. And we can also use the optimum populations for the different climate conditions. So you can see there's a great potential if we use if we do the climate match for at the population levels. This universal response function has been used in many cases, and we together did a study with Douglas Fir in Austria, and we built this function and predict the performance and to determine where is the suitable seed source. Now then we apply this one to predict entire Europe. In many cases, the prediction is pretty well, although the function is only developed based on the results from Austria and the part of Germany. And additionally, we also use this one to predict the distribution of the native in the native range in North America. Here is the distribution occurrence of Douglas Fir. Here, if we use the occurrence data from Europe to predict the presence in North America, we can only find these are only from the coast. The many parts are missing. But if we use the URF, whether to use height or basal area, so the prediction is much closer to the real observations. And very recently, Lippi and others used the universal response function to define the plasticity and the local adaptation. And later on, after the URF is developed, later on, latest proposed universal transfer function using linear mix effect model. So the objective is to overcome the problem of small number of test size. You know, the URF require a reasonable number of test size, but for their case, they have only a small number of test size. They also trying to introduce the random effect to improve the model accuracy. So the predictor include transfer distance, that's why they call it transfer function, and the provenance climate. And this together actually can determine the test site climate. Then the interactions between the transfer distance and the provenance climate. Then they include some random effect of provenance, site, and the block. So we tested this UTF with lodgepole pan data, and we found that the URF and the UTF have exactly the same performance if random effect is not included. Now, do we need the random effect in it? Although this is different, the URF, you can see that has a provenance climate, site climate. The site is, both are explicit. We can see what is the site climate, what's the provenance climate to determine the response. But in the universal transfer function, you don't have the site climate, you have a transfer distance. So the site is not clear. It's a kind of difficult, a layer of difficulty. So the random, another thing is the random term increase the model accuracy, but not the prediction accuracy. Because when the model has a random effect, when you predict for the new conditions, the random effect have to set to 0. So the random effect is not used in predicting, in projection for the future climate conditions or for spatial predictions. The only can be used for the specific test sites. Another thing is the random terms are expected to improve the parameter of the fixed effect. And the purpose is to overcome the issues of small population, a small number of test sites, and improve the prediction accuracy. So for that, we tested under various number of populations and also the size. And we found no clear advantage we found in our case with lodgepole pan data. So the inclusion of the random terms often lead to the model non-convergence issue, resulting in using a single climate variables in the case the lattice and also the one did for the oak in Europe. And using a single climate variables sometimes can be problematic. For lodgepole pan, if we use the mean annual temperature for both provenance test sites, we can have a very high R-square. However, when you look at the spatial pattern, you can see that it's misleading. The productive area is not in the real location. And after we add more climate variables, then we got reasonable spatial distribution patterns of the productivity or height growth. So the trade-off between including random terms and limiting the number of climate variables in the universal transfer function is a concern. So, and the factors limiting the climate niche model to predict the fundamental niche model is that the occurrence data is affected by interspecific computations, the physical barrier, and the binary output. So in order to predict the fundamental niche model, we use a URF which can overcome all this, because the common garden experiment doesn't have a interspecies competition. We can overcome the physical barrier. We can use the probability as output. So we predict the fundamental niche at a global scale for lodgepole pond. And it matches well with most of the observations where they have a successful introductions. So I'm running out of time. Okay, let me quickly go to the landscape genomic approach. You know, the genomic information is rapidly accumulating. So, however, the genotype-phenotype association is remain a challenge. And although the genomic prediction has some promising results, and anyway, here I'm showing results from landscape genomics to use for seed zone delineation. So it's essential to have the delineation for seed and breeding zone is essential for seed sourcing and the breeding programs. For lodgepole pond, we have different number of seed zones from different studies. And the principle of the seed zoning zonation is that we need to minimize the within zone variation, and while we still have a reasonable number of zones to manage. So it has been done based on common garden experiment, which are, expensive, long-term, and not sufficient for many, or even not available for many tree species. So there's also ever-lasting debate how many seed zones we should keep. So we use the gradient forest model with a large number of snippets from adaptation project. We use the climate variable scale-free climate data from Climate AA, and we can define the the species spatial genomic variation for the species in BC and Alberta. Then, based on the spatial genomic variation, we split it into different number of zones. We look at the change of within zone genomic variation. Is reducing with the number of zones increasing. But we don't see a clear turning point. So then we also calculate the reduction of the within zone variation. We found that here is a clear cut. So we determined that 6 zone is the most logical number of zones. So we compare our zones with the earlier studies. We found that 65% agreement with Lippy's study, but we found it shows some advantage because the Lippy's method has clear impact from the political boundaries, and ours are independent of that. We also compare with another zone based on the genetic correlations. And they have 4 zones, we have 6, but we found we can separate the clear difference between the interior and the coastal climate. Okay, in summary, the genetic Climatic mismatch at the species and the population level is the key of the most problems caused by climate change. There are many problems, but this is one of the key issues. Nature has created many species and genetic lines for different climate conditions. It's just a matter how we use them in a way. Climate niche model provide effective tools for climate matching at the species level, and the transfer and the response function can be used to help the seed sourcing and the seed orchard planning at the population level. And the fast accumulating genomic information has a great potential to serve as an alternative to our conventional approach. Here is my group. Everybody is working on climate matching at different levels from different aspects. Here are our sponsors, and thanks a lot. Sorry for the overtiming. Any questions, please?
Thank you very much for an excellent talk, Tongli, and for taking the time to join us from Canada. I'm sure there'll be some questions, so I'll just open the floor. Would anybody like to kick off? Catherine.
Thank you, Tongli, for this nice talk. I have a question about how we can learn from a comparison of climate niche modeling of the different species.
Okay, how to compare climate niche modeling across different species?
Not only comparing, but what kind of information we can learn from these comparisons of modeling between species, which could be helpful to complete the view for each of the different species. Could we consider that comparison between species could be more informative than just regarding climate niche modeling from one species independently?
Sorry, there are some part I didn't get it, but let me repeat your question. How can we learn from modeling different species to see? There are something I didn't get.
Yes, it's just could we learn from modeling in different species to improve modeling for one species?
Oh, I see. I never thought about that. We model many species. Each species has their own features. And some species is easily to model. It has a reasonable result. Some species is different. And you need to really balance the absence, the proportion of absence than the present data. It's hard to generalize. But anyway, we are working on modeling as many species as possible, then put them together. And traditionally, we are looking at from the species point of view. But I think it's important to, from the planting site point of view. When you have a planting site, you model all the species, and then you can make selections which species is the best for the given test size. I'm not sure I'm completely answer your question.
Okay, yes, I think that did. Thank you very much. Any other questions? If I could maybe ask one.
Sure.
I just wonder, do you think the use of the universal response function in overcoming the limitations of the numbers of test sites, but for a lot of species, might open up the potential to make use of a lot more sort of historic experiments that have been laid out that maybe don't have the geographic scale of some of the experiments you presented?
It can overcome to some extent, but still is the number of test sites need to cover at least a quite a range of the climate conditions. You know, your URF use both provenance climate and the site climate. If the so they can refer each other to strengthen the model, but this can help to some extent, but not if you only have less than 10 test sites cover a quite narrow range, that is still problematic.
I have a question, a very naive one, coming a little bit from outside. I'm not a modeler, but have you tried, or are you thinking, or do you know about people doing models using more than one trait, multi-trait models, that in a way that you could predict responses, not for just height, or but for an integrated phenotype?
Yeah. Sorry, you know, due to the audio effect, I have problem to hear from the question from the audience. Could you repeat it? I mean the
I was asking,
navigator. Yeah.
Yeah, I was asking. The models you have been showing in your talk are for one single trait. Like
Yes.
And I was wondering if there is also possibilities to do models for more than one trait together, like multi-trait model combining and like you know, to get to model a full phenotype. Combining.
Good question. Good point. We in our demo, the first studies, we model the tree height, and for the study in Austria, we also model the diameter or the basal area, and but we model them independently. We have never thought about combining different traits to make multivariate analysis for a universal response function. It can be complicated. I haven't thought about this to put them together. So far, my suggestion is to model them independently, put them together, or say you can model the volume instead of height and the diameter. The volume is an integrate trait. It's a good point. You can explore it.
You presented an interesting slide about the number of provenance trial needed to have an accurate model. And the curve was decreasing a lot, down to 40 sites, as far as I remember. So it means that if we want to install common garden or provenance trial dedicated to adaptation, if we have 40 sites covering various climates, this would be enough to set up accurate models.
So the model I show, the results show is for a number of populations. So it's a common, many provenance trials have several tens populations. For the number of test sites, yes, the limitation of test size is the issue. You know, the random effect seems when your number of population or size very small, it has a slightly better performance. But the problem is the non-convergence problem. When you have the random effect in the model, you cannot include more climate variables. So that's a challenge. We really try to, between the transfer distance using transfer distance and using the provenance climate, we prefer our URF over UTF. However, I thought that introducing the random effect is a good idea. We tried different ways, but the results is not encouraging.
Anyway, I guess that's a random effect will make the black box for the part of variation you can't explain. If you have enough sites to include more than one climate from the site, then it might be explained and not need anymore to this random effect. But I had another question. I was discussing this issue with Delineation models. They say, why don't you first do a high growth function and then expand the factors, ratio or whatever, and then you get 2 or 3 parameters of this function, then you expand it by the climate factors. That would might be another way to make one adjustment for 10 years and one for 15 and one for 20, but to model it.
Sorry, I can barely hear your words. I don't know. The audio effect from audience is very poor. Could somebody repeat?
Ouais, je me demande si ça serait pas bien.
C'est quel le micro qui prend?
I can try to repeat the question. Can you hear me clearly?
Yes, it's still not clear, but yes. Let's confirm every point I get. The microphone in the front is good, but in the audience is so poor.
Let's try here. I was discussing this with growth and yield modelers, and they say, why don't you first adjust the growth model, a high growth model, and then you get the parameters of the function, and then you can expand the function by site or climate factors. Then maybe you would get these parameters to be compared between sites and provenances or populations. And that might be a smarter way to study the phenomenon high growth than to do, let's say, discrete age adjustments when you have the problem that not all provenance are measured at the same age and so on. So do you think that might be a feasible approach?
Sorry, you said you develop the parameters for a site and the provenance first, right?
Yeah.
How to do that?
By high growth adjustment, so age and height, and then you adjust a function for high growth.
Oh, you mean the age adjustment?
Yeah.
Oh, if you have a provenance trials established at different years, so now you have different ages, how to combine them together, right?
Exactly.
Okay. Yes, we did a study in the Chinese Cyprus, and that's established different years. So we did the year adjustment first, then we develop your universal response function.
Okay, thank you. I think we have one more question, if that's okay.
Yes, I have a question about the possibility to be this kind of model self-updating. Well, with the URF or UTF, we can predict the expected performance or value, but we also have the interval of confidence of the model. So my question, if you believe that this kind of results could be also used to decide where new common garden experiment should be planted, maybe on the limit of the prediction or where the prediction is more uncertain.
Yes, that's a good question. You know, as I shown, for lodgepole pond, we have 60 test sites across province. And when we plotted against the climate, we found 20 of them are concentrated at mean annual temperature of 3. So that's a lot of repetition. We need to determine the climate conditions. Now we have climate data available, right? To pre-select the test sites instead of at the 70s, year 70s, there's no climate data available such widely. So they decide the test sites based on geographical distributions. Now we can use the climate model to predict climate conditions, then to determine where is the best place to put our test sites, and also the sampling of the populations. So the AMAT trials established recently, we need much smaller number of test sites and the populations.
Thank you. I think that sounds like a very good place to stop. I think that gives us good way forward for future work. And I'd just like to thank you again for taking the time to be with us and to answer a lot of questions. It was an excellent talk and I think a really good way to draw today to a close. So thank you very much.
Thank you very much. How to...
Okay, and I think that draws our session for the day to a close. So thanks everybody for all the contributions and good questions. I think it was a really interesting day. Look forward to more tomorrow.